Okay, let's talk about mutations. Now, mutations can be changes in the base sequence of the DNA. Of course, the base sequence controls the amino acid sequence, so any change in the DNA sequence will affect that, which may then cause damage to the cell, or may even lead the cell to be non-viable and die. Okay, so here we've got, this is supposed to be a DNA replication fork. So we've got DNA here, and DNA polymerase is working in this direction here, in the 5 to 3' direction. And right, so G, what happens is the complementary base of that is C, of course, and then we've got T, A, G, T. So all those nucleotides are going to line up against the complementary base, and then the DNA polymerase will come along and it will knit those together. Right. Now let's think about the different sorts of mutation. Let's think about chemical mutations. So certain chemicals can cause mutation. So let's think, for example, the substance benzopyrene, sometimes called benzoapyrene, and that's present in tobacco smoke, and that will react with guanine residues. Okay, so it may react with that one, and it makes it very bulky, and then means that it's not going to be it disrupts the structure of the DNA. That can cause a few different things to happen. So it means instead of getting a C there, you may just get an A or a T, or even another G, sort of another base put in there which doesn't belong. Or you could it can cause a deletion, so you might not get any anything pairing there, and the next one then will be that A there, so you missed one out. So you can either change a base, substitute it, or it can cause it to be deleted, or it can cause another base to be inserted there. So for example, you might end up with, you're not supposed to have 2 there, you've even inserted an extra base. So that's how chemicals can do that, and different chemicals can react with different bases in DNA and cause these sort of problems. Let's talk about UV radiation. Well, UV radiation causes the generation of reactive chemicals called oxygen-free radicals. Okay, free radicals, which can then go and react with bases, just like a chemical mutagen. They can change the base, stop it pairing, so you get errors introduced there. X-rays and gamma rays are called ionizing radiation. They cause again, they cause the generation of free radicals, which then go on to damage the bases. And also some mutations are just spontaneous, nothing causes them. Okay, so DNA polymerase is an enzyme which is working along down here. It doesn't make many mistakes, and in fact it has a proofreading, so facility as well. So if it does make a mistake, it can correct it, but nevertheless sometimes mistakes do occur. DNA polymerase may insert the wrong base, or it may leave one out, or insert an extra one. So all of those things can change the base sequence in the DNA and cause a mutation. Right, how does that, we're going to primarily affect mutations which affect the coding region. So we've talked, you're probably aware that a lot of DNA doesn't actually code for proteins, but let's see if we get a mutation in the coding region, what can happen there. Right, so if we think about the 3 different things that can happen, you can have an insertion of a base, you can have a deletion of a base, or you can have a substitution. Right, let's talk about these insertions and deletions. Now, these are going to cause big problems called frame shifts. These are frame shift mutations. So if I just put a random sequence of, let's say we have a bit of a base sequence in our protein here. Let's say we've got G, A, T, T, C, A, G, T, T, C. It's made that up, okay. Now we know that the genetic code is read as a series of triplets. So that GAT is going to code for one amino acid, TCA another one, GTTC the third amino acid. Let's make a fourth one. So you've got that sequence will code for 4 specific amino acids. Now let's say that we get this. Let's say that this T here so this T is deleted. Okay, now that's deleted. It's going to change. It's going to mess up everything. So the first triplet that's going to be read is that, and the second one is CAG, which is completely wrong, and then that's completely wrong as well, and so on. So everything after that deletion, everything after that deletion, sorry, everything after that will be incorrect. That's what's called a frame shift. So it's reading the codons in the wrong frame, and so that will lead to right. You go frame shift. So you get all the wrong amino acids. So you get a completely non-functional protein. That protein doesn't do what it's supposed to do. Now imagine if this happened in a very crucial gene, like for example the gene for the beta chain in hemoglobin, and well, in a fetus, the fetus would not be able to make beta globin. It wouldn't have hemoglobin. It would be non-viable. It would die. Okay, so of course you can get more subtle mutations in less essential proteins, which may not cause the cell to become non-viable and die, but can cause other problems. Of course, some mutations are also advantageous, and that's the whole mutation is the whole process which drives evolution. With no mutation, you would get no evolution. Right. Let's think what would happen if you got a you can see if you got an insertion there. So instead of putting that T in there, let's say we put in an extra. Let's say you put an extra C in there. Well, that's going to have a very similar effect. I just went there. So the codons of green, GAT is going to be red, then TCA, that's correct, and then GCT, that's incorrect, and then this one's going to be incorrect as well. So everything after the insertion there is incorrect. You get a completely non-functional protein. Okay, now let's talk about substitution. That has a less catastrophic effect immediately. But let's have a look at a good example of that. And a really good example of that is the disease sickle cell anemia. Which is a disease, an inherited disorder of hemoglobin, of the beta chains in the hemoglobin. Now, in normal hemoglobin, you have the sequence and you have a triplet code GAG, which will give you the amino acid glutamic acid. So that's glutamate there. Now what happens, you get a single mutation of a base. The A gets, in sickle cell anemia, has been converted to a T. So that means now the codon is going to be GTG, and that doesn't code for glutamate. That encodes for the amino acid valine, which is quite different to glutamate. It's much more hydrophobic and it changes the properties of the protein and causes the disease. So that's a good example of a point mutation where you have substituted one of the amino acids for another one, and that causes well, it's caused by the base substitution, which causes an amino acid to be substituted. Another kind of substitution, you can end up getting a truncated protein or a short protein, and we'll see how that happens. Okay, so for example, there are 3 stop codons in DNA. So for example, UGA in the RNA it's UGA in the RNA. So that means the corresponding sequence in the DNA, of course, you don't have U in DNA, you have T. So it's going to be TGA is a stop codon. Now, very similar to that, TGA, you could have TGC. TGC is codes for the amino acid cysteine. So this is stop, and this is cysteine. So imagine you should have a cysteine residue. You're coding for a cysteine, and you're going to see some kind of mutation event occurs, which changes that C to an A. Now the protein, instead of having the cysteine, it will stop translation of the protein. So maybe this is the correct protein, and there you've got your cysteine residue in there, let's say, cysteine. If you get the mutation, the protein, well, the protein synthesis will occur correctly up to that point, and then it will stop. So you get a short protein, a truncated protein, and that obviously is not going to work. And a good example of that is in the disease in cystic fibrosis. There are lots of different mutations. There are over 100 different mutations which can cause cystic fibrosis. But in some of them, you get one of these point mutations which converts a codon to a stop codon. So you get this truncated protein, which is non-functional, and so the person suffers from the disease. So anyway, there's just a few different ways in which mutations can lead to changes in coding regions of DNA. Mutations which occur to non-coding regions sometimes have very little effect at all, or sometimes they do have effects, much more subtle. So quite often, if you're going to get a mutation to a really key protein, such as hemoglobin, obviously that's not going to have a subtle effect at all. Very often the organism will just be non-viable and won't even survive, whereas maybe mutations in non-coding regions have less dramatic effects, which may affect the organism in different ways.